Welcome to Planetterrian Daily, episode 122, coming to you on July 16th, 2026. Here's your science and health briefing. A preprint shows amino acids can form replicating peptides and cell-like structures without nucleic acids or lipids. A bio-RXIV preprint reports that amino acids spontaneously polymerize into peptides that template copies of themselves. The resulting structures exhibit longevity and catalytic activity in the absence of nucleic acids, lipids, or sugars. Proteomic tracking showed sequence evolution over time within the peptide populations. The authors propose this pathway as a plausible bridge between Miller-Urey chemistry and early functional biology. Further experiments will quantify replication fidelity under varied environmental conditions. The bio-RXIV preprint demonstrates that amino acids can assemble into peptides capable of templating their own replication and forming cell-like structures without nucleic acids or membranes. This observation supplies a concrete experimental link between prebiotic chemistry and the emergence of functional polymers. It moves the field from theoretical scenarios toward measurable replication kinetics and sequence evolution. Next, the authors plan to quantify how environmental variables affect replication fidelity and structural stability. Most people assume life required DNA, RNA, or lipid membranes from the very beginning. Experiments now show peptides can form, replicate, and evolve without any of those components. Right now, inside every cell, proteins still perform the core catalytic work while nucleic acids mainly store information. The new findings suggest this division of labor could have arisen gradually. A single round of peptide templating produced measurable sequence changes within days under laboratory conditions. The practical takeaway is to watch for future studies that test whether similar peptide systems can persist and diversify under realistic early Earth conditions, such as wet-dry cycles or mineral surfaces. From the earliest molecular systems that could copy themselves, we turn to a modern microbial mechanism that may drive disease once those systems are in place. Researchers identified how a bacterial toxin linked to colorectal cancer first attaches to the receptor Claudin-4. This binding lets the toxin disrupt the protective epithelial barrier and trigger inflammation. In mouse experiments, a decoy protein blocked toxin access and prevented downstream damage. The work clarifies a precise entry point for a long-suspected microbial driver of colon tumors. Follow-up studies will test whether targeting Claudin-4 can reduce tumor incidence in higher-risk models. While one microbial interaction can undermine a protective barrier, large-scale genomic resources are now revealing how genetic variation shapes disease risk across entire populations. The NIH program now holds the world's largest integrated set of genomic and lifelong health records. Participants have contributed both whole genome sequences and comprehensive electronic health data. Researchers are using the resource to map genetic contributions to common diseases across diverse U.S. populations. The dataset continues to expand toward the original target of 1 million volunteers. Analyses of rare variants and ancestry-specific risk patterns are already underway. A new computational method improves identification of genes that define cellular states from messenger RNA-seq datasets. The approach was tested across multiple tissue types and disease models by researchers at IGTP and UPC. It yields more interpretable gene lists than prior tools. Integration into existing analysis pipelines is underway. Large human datasets illuminate developmental differences across species. Next, we look at how reproductive strategy altered the timing of limb formation in marsupials. Scientists found that marsupial arm development begins well before birth and bypasses the typical limb-building sequence seen in placental mammals. The accelerated timing supplies newborns with functional forelimbs needed to reach the pouch. The study provides new insight into how reproductive strategies shape developmental timing. Comparative gene expression maps highlight regulatory changes that drive the shift. Future work will examine whether similar accelerations occur in other marsupial organ systems. A primate with pinkish-orange lips and a black face has been formally described as a distinct species. Field observations and genetic sampling distinguished it from known congeners in the Democratic Republic of Congo. The discovery adds to the documented primate diversity of the region's forests. Taxonomic work continues to clarify its range and population status. Analysis of the 2021 Marshall Fire showed residents often traveled to places with existing social ties rather than the nearest safe location. The pattern held across multiple weeks of displacement. Researchers link the choices to the strength of prior community connections. The findings suggest evacuation planning should account for social geography as well as physical distance. From developmental timing shaped by reproductive needs, we move to collective behavior that helps single-celled organisms survive under pressure. Queen Mary University researchers observed marine Cyotabinacea aggregating into dense groups when grazed by protists. The collective behavior reduces individual loss and may alter carbon export rates in surface waters. Laboratory and field data link herd formation to changes in local carbon cycling. The strategy reveals a previously unrecognized microbial defense mechanism. A review details how excitons generated by light interact directly with magnetic order in two-dimensional quantum materials. These interactions allow optical control of magnetic states without external fields. The materials exhibit responses not observed in bulk crystals. Potential uses include light-written memory and low-power photonic logic. Device prototypes are now under development in several labs. A donut-shaped topological framework maps how yarn connectivity produces fabric properties such as elasticity. The model distinguishes stretch behavior arising from knotting patterns versus fiber composition. Researchers validated the approach across knitting, crochet, and woven samples. The classification offers a design tool for engineering textiles with targeted mechanical responses. Trinity College engineers produced a geopolymer formulation that prints without Portland cement. The material uses industrial byproducts and demonstrates structural viability at scale. Tests confirm compatibility with existing 3D printing hardware for construction. The work supports circular economy approaches to low-carbon building. Uppsala University scientists described a root-associated fungus as the first member of semicentennial The species Semicentennalia rex honors the monarch's 50th anniversary. Morphological and genomic data place it outside existing fungal classes. Further surveys will determine its distribution and ecological role. Cambridge researchers calculated that increasing the tournament to 48 teams raised total emissions to 4.23 million metric tons. Audience travel accounted for 82% of the footprint, with flying responsible for roughly 3 million tons. The study estimates that similar mega events could cut emissions substantially if organizers offered discounted tickets for closer venues. The findings quantify the dominant role of spectator transport in event-related carbon costs. Attribution analysis shows extreme rainfall events in the region are now 5 times more probable than in the late 19th century. The study examined last month's floods that killed nearly 100 people. Model comparisons isolate the contribution of human-driven warming from natural variability. The results underscore rising regional flood risk under continued emissions. Microbial strategies for persistence lead us to a simple chemical route that could help manage the persistence of plastic waste. An international team showed that tiny water droplets plus oxygen can break down recalcitrant plastics into high-value organic acids. The reaction occurs at ambient conditions without added catalysts. Yield and selectivity data indicate practical potential for decentralized plastic upcycling. Scale-up trials are planned to assess throughput. Before we go, keep an eye on scale-up trials testing water microdroplets for breaking down plastics into organic acids under ambient conditions. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at Nera Network. Links in the show notes. And before you go, this show is part of the Nera Network, a family of daily podcasts on tech, science, and markets, each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. Tesla Shorts Time, your daily deep dive into everything Tesla. Find every show free at naranetwork.com. The age of AI puts real builders on the phone with an AI host. Apply at naranetwork.com/age-of-AI apply. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.